And Marlene, we do have the chat disabled, but people will still be able to ask questions through the Q&A. So there will be time for questions at the end of the presentations, which is great. Okay, so let's go ahead and get started here so we don't run out of time for our presentations. So for anyone who doesn't know me, my name is Brad Peter. I'm the Executive Director at the Alberta Lake Management Society. And we have two panelists helping us today. We have Janine Higgins, who I don't think her video is up right now, and we have Jenna Curtis, both through Alberta Environment and Parks. They're helping us with the technology side of the webinar series this month. So thanks to both of you, and they'll be helping facilitate questions after each of the presentations. Obviously, I wish we could be doing this in person this year, but we're trying to make the best of a bad situation, and we've invited great speakers from across North America to present on a variety of lake topics. So hopefully that is, you know, an okay replacement for our usual two-day in-person conference. And today we're starting with two topics which are a little bit closer to home. We're starting with Caleb Sinn from the Alberta Lake Management Society, and then around 11 o'clock we'll be hearing from Nicole Kimmel from Alberta Environment and Parks. And as I just mentioned, the chat function is disabled, but you will still be able to ask questions through the Q&A function. So throughout the presentation, feel free to type questions as you think of them. And Janine and Jenna are going to help facilitate those questions to the speaker at the end of the presentation if we have time for any of those questions. So I think without further ado, we'll pass it over to my colleague Caleb at the ALMS office, who is going to start us off with the first presentation.
Thanks, Brad. Hi, everybody. I'm just gonna start sharing my screen here. All right. So yeah, as Brad said, I work with the Alberta Lake Management Society, and I'm going to give a little program update about what we've been up to this summer. If for whatever reason you can't see my video, that's me. I've been the program coordinator with ALMS since February. And before that, I was a lake watch technician in the 2019 season. And if you're not familiar with ALMS and what we do, we promote the understanding and comprehensive management of lakes and reservoirs and their watersheds within Alberta. We are a nonprofit, started in 1991, and we primarily undertake education initiatives through seminars, conferences, webinars like this, and also lots of one-on-ones. We also collect, synthesize, and report on lake water quality information. We provide scholarships to foster aquatic research in Alberta. We provide expertise to organizations also concerned with lakes and watersheds in Alberta. And we participate in local, provincial, and national programs pertaining to lake reservoir water quality as well. So I'm just going to give a quick overview as to what I'm going to talk about. On the left of the screen is a map of all the places that we sampled this summer. I know it looks kind of hectic, but as I go through the presentation, we'll split up the map and you'll be able to see it a bit more clearly. I'm going to start with our Lake Watch program, which is our oldest monitoring program, then go on to lake keepers, and then our satellite monitoring of cyanobacterial blooms, and on to what we've been doing this summer with eDNA analysis and monitoring. And then I'll talk about Eurasian water milfoil monitoring and other things that we've been doing with aquatic plants. And then I'm going to wrap it up with our scholarship recipient. All right, so Lake Watch is our participatory science lake monitoring program, and it's participatory because we hire technicians that are trained to government standards to sample lakes, and they work with volunteers across the province to sample their lakes. So the way it works is the technicians will coordinate with these volunteers to sample 4 times across the open water season from June till September. They take samples across the lake at one location. They do a profile to get physical and chemical information from the top of the lake to the bottom. And then they also collect samples across the lake and put into a composite, which we get analyzed for various chemical and biological parameters. And when we get the data, we upload it to the government database, and then we also report the data through our reports that we publish online. So the animation on the left is all of the locations that we've sampled since 1996. And last year, the season that I was at Tech, we were able to sample 26 different lake basins with the help of 48 volunteers contributing over 400 volunteer hours, culminating in 104 lake monitoring trips, which is about 93% success rate, which is pretty good given last year was a bit iffy with the weather. It was a bit cold and a bit stormy and windy. And the figure below is the average total phosphorus concentration for each of the lakes that we've sampled. So you can see we have quite a variability of lakes across the province that we sample. This year, we're sampling 24 lake basins through the Lake Watch program, which is about 96 planned trips. And we're able to do it this year with the support of Alberta Environment and Parks, the Pigeon Lake Watershed Association, Lacombe County, and LICA. And admittedly, we wanted to do more lakes this year, but given the COVID pandemic, there was some budget restrictions, but we're still more than happy to be able to get out to 24 lakes. That's still a very good program given what kind of happened or what's been happening. And these are our two technicians that we were able to hire. Ryan and Kira, they're really great, very easy to work with. They both have a good background in aquatic ecology through school or through prior experience. And this is what we're seeing out there. So Ryan and Kira are lucky in that the Parks Lakes that they get to sample this year, because we have a set of Parks Lakes that we rotate through every year. The Parks Lakes this year are mountain and foothills lakes. So on the top left is Lower Kananaskis Lake, and the top right is Beauvais. Those are some of the more nice, deep, clear lakes that we get to sample this year. And flipping on the other side of the spectrum, we have more the typical central Alberta lakes, Lac Lenon on the bottom left, Moose in the middle, and then Shining Bank on the bottom right. Yeah, so this year, as you may have noticed, it was a bit more sunny and warm. So we're starting to see the impacts of that weather on these lakes in terms of cyanobacteria, and lakes this year have been very green, and the blooms have started fairly early. And an impact of that, or what's going on this year, is lots of cyanobacteria advisories posted by Alberta Health Services. You may have heard about a dog that died at Moose Lake, likely because of exposure to blue-green algae or cyanobacteria. So just a reminder to everybody attending the webinar, if you have a dog and you're going to lakes this summer and you see cyanobacteria in the water, do not let your dog go in the water. Yeah, the results, as we've seen at Moose Lake, can be fatal. But despite that, the Moose Lake Watershed Society is doing a lot to understand what's going on in Moose Lake. And this year they're doing an enhanced sampling program, and we're helping them out with that to follow up on their enhanced sampling from 2017, where they're doing specific sampling at specific basins across Moose Lake. So this year we're sampling Franchier Bay and Main Bay. So we're getting specific nutrients from each of those basins. And at the same time, the Moose Lake Watershed Society is also calibrating a chlorophyll probe that they have purchased. And they'll be able to use that probe to get in situ or in water measurements of chlorophyll A to get estimates across the lake and even in the inflow and outflow streams for cyanobacteria. So we've also observed a few lakes this year having late June green algae blooms. So both Lac Santé and Wabamun stewards out there contacted us and said, hey, we have we have a bloom, and they sent us pictures, and they sent us some samples as well, and we were able to confirm that it was in fact green algae, which is distinct from cyanobacteria. The blooms are not toxic, but they can still be stinky and a little bit disruptive. So you can see at the docks at Lac Santé, the stewards there had to do a lot of cleanup so they could access the lake. And you can distinguish green algae from cyanobacteria primarily by its structure. So you can see the picture from Wildman Lake, the steward there is actually able to hold the colony of green algae, where with cyanobacteria, when it grows very, very densely, you can't really hold it like that. It's more scummy. But then at both of these lakes, later in July, they both demonstrated some cyanobacterial blooms, and they look a little bit different. Wildman, you can see, was a bit more brown. Lac Santé is a more typical green. It's interesting, those are two lakes that have moderate levels of nutrients, or what we would say mesotrophic. And some of the stewards out there are saying, hey, we haven't really seen blooms like this within the last like 5 or 10 years, and they're kind of wondering what's going on. And it's really important to continue to do the Lake Watch program so we can try to answer questions like what's happening, what's changing in the lakes. And that's why it's important to keep doing Lake Watch sampling so we can achieve long-term trends, like here at Laurier Lake, where we're able to say if we have enough data over a long enough period of time, that yes, there are significant increases in chlorophyll A or phosphorus, or decreases. So yeah, that's kind of the goal with the Lake Watch program, is to capture how the lakes are doing in the province, and to be able to detect if there are any significant changes in parameters over time, and then be able to use those findings to help with management initiatives. All right, on to our Lake Keepers program. This is our citizen science program, and we distinguish that from Lake Watch because we actually empower and enable volunteers across the province to do the lake sampling themselves. So we provide training resources online on our website, and then we give sampling equipment. And last year in the summer Lake Keepers program, we were able to sample 8 different lakes across the province. You can see from the map that they're pretty spread out. That's because the Lake Keepers program is designed to sample lakes that are a little bit more remote in the province, otherwise not all that easy to sample through our Lake Watch program. And the Lake Keepers program is also designed to sample lakes that don't have a lot of, haven't done a lot of sampling in the past, in order to get a bit of a baseline data for those lakes. Bit of an update for our Summer Lake Keepers program this year is that we've started measuring profiles with a YSI ProDO probe. That's a really big enhancement to the data that we can get. In prior years, we were using a Van Dorn Kemmerer discrete sampler to collect water at 3 locations through the water column to estimate what the temperature profile would be. As you can see, it's very coarse. Probably would miss the thermocline depending on what the middle depth is. So upgrading to using probes in this program has been really, really great. And another thing is that we can actually measure for oxygen, which gives us a lot of information about the chemical environment in these lakes. That's a big update. So Lake Keepers this year, we have 7, what I'm gonna call base Lake Keeper lakes. And I call them base because those are the lakes that are being sampled as part of the regular program, where they're sampled 3 times through the open water season, starting in June and going all the way till, we'll probably get samples in early October. And these lakes are sampled just at 1 location, generally the deepest part of the lake. Profiles collected at that location, and then surface samples are taken for chlorophyll A, nutrients, eDNA, and microcystin if a bloom is identified. And the lakes that are part of the Lake Keepers program this year are, we have a lot in the Peace watershed. We have Hague, Peerless, Graham, Swan, and Snipe that are a part of that group. A huge thanks to the Mighty Peace Watershed Alliance for coordinating the sampling in the region and finding volunteers to sample those lakes, as well as the Alberta Conservation Association, which is doing a lot of sampling through this this program as well. And then we have Gadsby Lake in central Alberta near Red Deer, and then Westover, which is a small lake just west of Calgary. And then Sturgeon Lake has a bit of an enhanced program because it's doing the regular sampling plus some more sampling around a new water treatment plant, and TSAG is doing that sampling. Then we have 5 other lakes that have currently been sampled as well through the Lake Keepers program. These lakes have only been sampled once. They are part of a project that Dave True, who is a board member of ALMS, is kind of heading up. These lakes are all in the Carvel Pitted Delta, which is just west of Spruce Grove. They're all unnamed lakes for the most part, except for Roy, which is commonly known as Gerhardt's, and they all are more or less on private property, and there's a really interesting diversity of lakes in that area. You get some lakes that are as deep as 15 meters and have really, really good clarity. And then just a couple of kilometers away, you have really shallow lakes that get really green. So it's really neat to be able to capture the diversity of lakes in that area. And the project also includes looking at the groundwater impact on those lakes, because Dave True is engaging some hydrogeology labs at the university to be able to understand maybe there's a really interesting diversity of lakes in that area because of some interesting groundwater impacts. And Idaho Lake is getting a bit more of an enhanced sampling this year. It's being sampled more like a Lake Watch program, just to get some more high frequency, high quality data that we can use to answer some interesting groundwater questions. So what we do with the Lake Keepers information when we receive it at the end of the season is we compile it into a report and publish figures like you can see on the left. All of our past reports are available on our website. And then we also upload the information to the Gordon Foundation's data stream database, which is an open source, publicly available hub for water quality information across Canada. It's really neat. So if you're curious about using our data, it's all available to download on the data stream. All right, moving on to some more of our special projects for the summer. We have a cyanobacteria monitoring using satellites program this year. This program started from some conversations with the Pigeon Lake Watershed Association and their technical advisory committee. This last winter, they were thinking of projects that they could do, and Dr. Rolf Feinberg thought, hey, maybe it would be a good idea to start monitoring cyanobacterial blooms on Pigeon Lake using satellites. It's a common thing used in other parts of Canada and across the world. And so from that idea, the group engaged the Alberta Biodiversity Monitoring Institute, ABMI, and because of their geospatial expertise, they were able to map out how cyanobacterial blooms have developed at Pigeon Lake in 2019 and 2018. And from the information that came from that, the group decided it would be a good idea to do validation of cyanobacterial blooms and chlorophyll levels at Pigeon Lake this year. And that's because we used models from lakes in Ohio and Lake Winnipeg, I believe. And it's important to use a model from the system that you're looking at, because lakes are very diverse. Their phytoplankton ecology and chemistry can really impact the imagery and in turn the chlorophyll levels. So that meant that we needed to get onto Pigeon Lake multiple times across this open water season at the precise on a day where the satellites were going over, and we did sampling at 20 sites. We collected water samples that are gonna be sent or were sent to the Weinberg lab for pigment analysis, primarily for chlorophyll A, but also we're gonna get some information for some accessory pigments that might be more useful for identifying cyanobacteria specifically. And we're also collecting samples for phytoplankton, which the Weinberg lab is also identifying, and we'll be able to relate what we're seeing from the satellite and the pigments with the phytoplankton community. And these images are the days that we have sampled Pigeon Lake so far. You can see that there's quite a development from the beginning of June till our most recent sampling on August 17th. The lake has definitely gotten much greener over time. So it's good to get the lake from over time in the summer to be able to get a really good model and a diversity of chlorophyll levels. And we also wanted to try to tack on a few other lakes on this project, because we know that a model like this would be useful for more lakes in Alberta, because there's other lakes in Alberta that are concerned about blooms and are used heavily or have towns on them. So we were able to do a few samplings at Wabamun, Moose Lake, and Lac La Biche. That picture of Lac La Biche is when we were on Lac La Biche, the East Basin, taking samples for this validation project. And the circle on the map there is where this picture was taken. It's just on the west side of the Sir Winston Churchill, the road to the park. And yeah, it's just, this picture demonstrates that there can be quite a variability and heterogeneity of cyanobacteria across the lake, which you wouldn't necessarily be able to capture in with traditional lake sampling programs, but that you can really capture well with satellite imagery. So yeah, just along with that, a big thanks to the Pigeon Lake Watershed Association for being so flexible with sampling. It's quite a challenge to be able to plan to sample a day that the satellite is going over. It has to be clear conditions, and you can't have a lot of wind because you got to stay safe out there, and the volunteers to be available. So it's pretty amazing that we were able to get at least 4 good samplings at Pigeon Lake, and one good one at Lac La Biche, and a few other good ones at Wabamun and Moose. So yeah, thanks to Lac La Biche County as well for taking us out on Lac La Biche to be able to collect those samples. And ultimately what we want to do with the information is potentially develop a tool similar to what Environment Canada is doing with the EO Lake Watch program, where they publish what the conditions are on the lake and through the season. And we're hoping that, yeah, a tool like this might be used by the public to help decide, where on the lake is good to go, or which lakes are maybe not that great to go to, or could be used for public health monitoring purposes as well. All right, switching gears again to our eDNA monitoring program. So this is our 3rd year doing eDNA analysis, but it's the first year that we're doing beach sampling. So the last 2 years we were just analyzing cyanobacteria genes. I think I went a slide too far. Yeah, here we go. We were analyzing cyanobacteria genes to quantify cyanobacterial levels and microcystin gene levels from Lake Watch samples. And we've been working with the Patrick Hannington lab to develop that program and get that information. And this year, as it so had it, we were brought in with Alberta Health to help with their beach monitoring program, because they were kind of lost capacity because of having to use their capacity to do COVID testing. So Patrick Hannington was in discussions with them and said, hey, ALMS is going to be at these locations or close to these locations anyways. Maybe we can just get them to sample. And we were more than happy to sample. And as you can see, we're able to get to a lot of places across the province and take these beach samples, as well as water samples from Lake Watch and Lake Keepers. And with that, we added some more targets for total enterococcus and a human fecal marker. And the sites, as I said, came from our Lake Watch and Lake Keepers lakes, but also from a priority list that Alberta Health provided us, primarily for enterococcus. And we didn't do all this sampling just ourselves, but we engaged our amazing network, and the North Saskatchewan Watershed Alliance took samples across their watershed, all the way west from Abraham Lake and east to the Whitney Lakes. Calling Lake was sending us samples fairly often. Lac La Biche County provided us samples from across Lac La Biche. The Summerside Residents Association was bringing us samples weekly from Lake Summerside. And the Pigeon Lake Watershed Association took a lot of samples from beaches across Pigeon Lake. So big thanks to all of those partners for being part of this cool project and bringing samples. And like I said, this program is possible because Patrick Hannington seeing the potential in our network and kind of going along with his research of thinking of ways to do decentralized monitoring for eDNA and public health targets. So thanks to Patrick Hannington for bringing us into this project and for providing us with our lab intern, which you can see there in the tree, Robert. He was very busy this summer because we got 123 beach samples from across the province and 92 pelagic samples, culminating in over 650 tests run. So yeah, Robert was very, very busy. Yeah, and just to wrap that up, this project was more research in nature as Alberta Health and the Hannington Lab is learning about how eDNA can be used to monitor public health targets. And yeah, so we're really interested to see how we can be involved in the future with more eDNA monitoring and yeah, just to continue that work. All right, so now onto our aquatic plants program. So this year, with the support of Nicole Kimmel, who's going to be presenting after me about invasive species in Alberta, we were able to collect specimens of water milfoil and submit them for genetic analysis to the Alberta Plant Health Lab. So the reason why we're doing it this way is because both Eurasian water milfoil and our native northern milfoil have very plastic morphology. They can look very, very similar. So the only really great way, the only good way to get a really good identification on these specimens is to get genetic analysis of them. So we were able to collect 28 different specimens so far from 26 lakes across the province. And thankfully, no Eurasian water milfoil has been detected. And all of them have been northern with the exception of one specimen from Brazo Reservoir was Whirl Leaf or World Water Milfoil. It's really cool to detect that. We've confirmed that it has a range within Alberta, a native range. So yeah, it was pretty neat to be able to detect a different species that we haven't been able to identify in the field. And yeah, we're going to continue to monitor for Eurasian milfoil because it's very abundant in BC. And a speaker as part of this webinar series, James Litley, is going to be presenting on Eurasian water milfoil in the Okanagan, I think in 2 Fridays. So if you're interested in water milfoil in BC, definitely tune into that webinar. And thanks again to our collection partners. The NSWA again was able to collect samples from across their watershed, and Lac La Biche County was able to collect samples from the lakes that they're sampling this year. So a big thanks to both of them. We did one plant bioblitz this year at Lacombe Lake. We sampled 50 sites, collected 106 specimens, 10 species were identified, and thankfully no invasive species were detected. We also have a new aquatic plant guide. It's our 2nd edition, and Brad poured his soul into this book, all of his life energy. So definitely check it out. It's really well designed. We were working with some really good designers to make this guide. It includes a complete list of prohibited aquatic plants in Alberta, along with some new native species and some lookalikes or comparisons of native species and invasive species. So you can purchase it online from our website. All right, and we also have selected a recipient of our ALMS scholarship. So Sydney Huckaluck was awarded $2,000, and her project is looking at nutrient limitation and co-limitation in agricultural streams across Alberta and how nutrient limitation impacts algal biomass and community composition. So definitely a really interesting topic for us in terms of how these streams are impacting our lakes. She's going to be presenting her research at our 2020 AGM this November. Will also be an online format. So if that topic interests you, definitely check out her presentation at our AGM. All right, so what's next for ALMS? What are we going to be up to? Well, we have Lake Watch lakes and Lakekeeper lakes that we're still sampling. So Kira and Ryan are sampling today. They're both, Ryan's at Lac Lenon and Kira's at Wizard. They'll be sampling until the end of the month. Hopefully they won't have to go on into October because they can get pretty cold on the lakes in October. We have our AGM in November to look forward to. I will finally get around to the Winter Lakekeepers 2020 report in the fall once our field work wraps up. And then we also have our 2021 Winter Lakekeepers program to look forward to. So if you're interested in sampling your lake or any lake in Alberta for winter information, definitely contact us. You'll be able to see my email at the end of the presentation. We will be working hard on our Lake Watch reports, our Lakekeepers, Summer Lakekeepers report, as well as some reporting for our EDNA program. We'll be working hard on our information from the satellite monitoring program and seeing what the next steps for that project will be. And then we will be involved with World Water Day in 2021 since it was postponed due to the COVID pandemic this year. And before I wrap up, just want to mention this spring. It was looking a little bit dicey as to how much we were going to be able to do this summer. There was a lot of unknowns, but we can really just have a lot of gratitude and thanks for everybody who was able to be part of our sampling this year and everybody who supported us. We were able to work with a lot of different government departments, a lot of great volunteers across the province and nonprofit organizations. We definitely wouldn't have been able to get to all these places across the province if it wasn't for people believing in our programs, supporting us, driving boats, putting bottles in water, going to Purelator, sending us samples. So a big thanks to everybody who's part of our programs. It really means a lot, and we were able to get a lot of good information from lakes in Alberta this summer. And with that, I will wrap up. So does anybody have any questions?
Okay, thank you, Caleb. We're just going to take a sec here to bring up the Q&A, and Janine and Jenna are going to help out with fielding some questions for you. If you have any questions, please submit them through the Q&A function through Zoom. We do have a few minutes here before next presentation, so throw your questions out there, and I'm sure Caleb would be happy to address them. Okay, I'll pass it over to Janine and Jenna.
Perfect, thanks, Brad. So we do have one question here asking Caleb, and thank you, Caleb, for that great presentation. If you can explain what Carvel pitted delta lakes are.
Okay, so generally they are, the Carvel pitted delta is a geological region that's just west of Spruce Grove. So yeah, the name just is to identify the region as to where these lakes are. And so it was a delta formed through glacial activity. And the lakes are generally very small, but are unique because they can also be very deep and very clear, which is surprising considering some of the size. So yeah, I hope that answers your question.
Hi, Caleb. Got your next one here. So first, they said, great presentation. Can you expand on the eDNA sampling methods?
Sure. So the collection is dependent on the program where the samples came from. So for our Lake Watch program, the sample came from our composite sample where water from across the lake was combined and then poured off into different bottles for different analysis. So the Lake Watch sample is collected like that. From our lake keepers program, it is just a vial that's filled at the surface at a single location. And then for the beach sampling, it is a composite across the beach. So 5 different samples are taken across the beach, and samplers are told to take a representative sample across the beach using 5 locations. And then that water we put onto a filter, and then the DNA is extracted from that filter. And then from that, we do qPCR analysis to get quantitative information for genetic information that we're curious about. And in this case, our targets were total cyanobacteria, a microcystin gene, and then total enterococcus and a human fecal marker.
Great, thanks, Caleb. So I don't see any other questions right now. So if anybody does have more questions, please feel free to type them into the Q&A and we will get to them as they come in.
Or if you want me to elaborate on any of your questions, then I can too.
Maybe I'll just mention to Caleb that the nice thing about this eDNA monitoring is that all of the samples, once they're filtered and extracted, you can go back into the archive samples and run them for any new different targets. So all of these samples that we have been archived, and you could run, if you had the right marker for it, you could look for zebra mussels, quagga mussels, genes for different toxins and things like that. So, you know, through the program, we're kind of building up this potential for future projects as well.
Yeah, and even looking for species at risk as well. It's really limited by what organism we want to look for.
Great. So we don't see any other questions coming in.
Okay. Do you guys want to maybe give everyone a 5-minute break? Run to the washroom, refill your coffee, whatever you need to do, and we'll get our next presenter, Nicole Kimmel, set up with her presentation, and we'll start with that in about 5 minutes here.
Perfect. Yeah, so it's 10:40 right now, so we can get started with Nicole at 10:45. So if everybody wants to take a quick break, like Brad said, please feel free to refill your coffee, take a stretch, do whatever you need to do, and we'll be back on it in 5 minutes.
Okay. And thanks, Caleb.
Yes, thank you, Caleb, for a great presentation.
Yeah, okay.
Yeah, go for it.
Okay. So our next speaker, as you can see on the screen here, is Nicole Kimmel, and she's the Aquatic Invasive Species Specialist with Alberta Environment and Parks. She'll be giving the next presentation for about the next 45 minutes. Again, if you have any questions that you think of throughout the presentation, please just throw them into the Q&A function on Zoom, as the chat function is disabled right now. And I mentioned a few minutes ago, the webinar is being recorded, so if you do miss part of it or your colleagues were hoping to tune in, we will be posting them on the ALMS website at some point, so just keep an eye out for that. So I'll pass it over to you, Nicole.
Thank you very much. So anytime I do a presentation, I never know how much folks have a background in our aquatic invasive species program. So there is a lot of repeat for anyone that's probably heard me before, but there's also some new tidbits of things that happened this year or happening. So try and stay for those little tidbits to keep you engaged. Our program is highly directed towards preventing zebra and quagga mussels from entering Alberta. Our closest location in Canada is Manitoba, and they keep finding new and new locations that creep closer to their Saskatchewan-Manitoba border. So it is getting closer and closer to us. The threat is very real. We suspect that we probably would be about a $75 million expense if mussels were fully established in Alberta. So this is huge economics, and it affects anyone that uses water, which is all of us. So it has huge impacts. But that's not the only thing that my program deals with. It's a bit of a hodgepodge of species that anything that's in the water that doesn't belong sort of sometimes falls onto my plate to deal with. So We've dealt with some tilapia being introduced into the Elbow River down by Calgary. They are a very warm water fish, and so we allow these for fresh food markets. But the risk is pretty low for them being actually introduced and surviving in our waters based on the temperature. So when they were introduced into the Elbow River, they probably all died, but we do have concerns about the diseases or viruses that they may have introduced in that as well. We got a call from the Calgary International Airport about some Chinese mitten crabs that were actually declared as TV lights. So we worked with them and found out that we had no jurisdiction on this, and it was actually the Canadian Food Inspection Agency that had jurisdiction over this species. These are my office pets. These are grove snails. They have been introduced in BC and doing very, very well there, but probably wouldn't overwinter in Alberta's cold winter temperatures, but was introduced into Alberta through some horticultural nursery samples. So it actually evaded inspections in multiple points and actually managed to find its way through. Crayfish are a bit of a native species, but definitely expanding in range. So we're trying to figure out the elements of spread on that. And I'm going to touch base on that a little bit further. This snail was the giant African land snail, also imported accidentally, but a person came from Hawaii and collected the shell, thought it was pretty, put it on their coffee table, and then overnight it had managed to climb across the coffee table. So they surrendered it to us, probably again wouldn't overwinter in Alberta. We kept it as a pet, and it can be quite devastating in warm temperatured climates, can eat stucco and crazy things, so definitely alarming. We did get a sample of an American bullfrog imported into U of A tadpole. They recognized that this was an off type, and they reared it up to see if they could identify what the tadpole was, and it did come out to be an American bullfrog. And last but not least, the showstopper, somebody did claim to have found an octopus down in a reservoir in southern Alberta. This came through Facebook, so we were never actually able to confirm it ourselves, but the picture did hold true with the snow in the background and the picnic table being a likely suspect for the location. So you never know what's going to come across my desk. So just to dive into the Alberta Invasive Species Program, we have 5 elements. I'm housed in the middle, and I beg, borrow, and steal people to actually coordinate all 5 elements. So we rely heavily on policy and legislation as a backbone to our program. We try to educate folks as much as possible on why we care about these species and what they need to do in order to change practices to prevent their movement and introduction. We work with Ron on monitoring, and Cindy Sawchuk does the inspection program. That is a huge program in itself, and she does wonderfully in leading that, as well as seasonal wage staff for the inspectors, and has 3 canine dogs under that program as well. And then once prevention fails, we have response. We have 2 routes, basically, emergency response and an ongoing long-term response, where we have to kind of take year after year of monitoring a species. So just to quickly go through policy and legislation, we've been building on this since 2015, where we did a huge overhaul of our Fisheries Alberta Act. That's when we went from 2 to 52 prohibited species, made those watercraft inspection stations mandatory, and then enhanced the ability to quarantine boats and even quarantine a lake if needed. We expanded that in 2016, pulled the plug, made it mandatory to report aquatic invasive species, recognizing that if we get notified as soon as we can, then we have a better chance of being successful in eradication or control. 2018, we launched the specified penalties, $324 for bypassing a watercraft inspection station, and $180 for failing to pull the plug. We've also got policy in place. So we have the Alberta Invasive Species Early Detection Rapid Response Plan. We've been operating in this plan since probably 2013, around there, in draft form, but it was just endorsed last fall in 2019. So this is fully publicly available, and it does have pieces in place for lake stewardship groups and municipalities for sure in that response. We also have the Decontamination Protocol, which was spurred by whirling disease. We quickly got government staff and contractors under government contracts to adhere to this Decontamination Protocol. And just recently, we've endorsed it for industry. So now industry has to also follow this Decontamination Protocol, which is basically based on the principles of clean, drain, dry at a minimum. And then as your risk goes up, you can add chemical treatments or temperature treatments to hold whirling disease at bay, as well as all aquatic invasive species. We have some proposed amendments underway under public consult for the general Fisheries Alberta regulations. One is to repeal the restriction preventing fishing for crayfish in the Beaver River and its tributaries. So at one time, it was believed that crayfish were only native to the Beaver River watershed and needed to be protected. Now that they've expanded in range, they are not, susceptible to eradication or extinction. So we are repealing that to open up fishing to hold their numbers at bay. The next one is we're adding a new definition for aquatic bait. Right now, there is no aquatic bait definition. So if we wanted to somehow mitigate that risk of aquatic invasive species being introduced in aquatic bait, it first needs to be defined. So first step is to get that definition in the regulations. And then the last one that affects aquatic invasive species is adding the live part to crayfish. So in section 8, it indicates that possessing live bait and crayfish is prohibited unless an individual is authorized to do so. So it was implied that live applied to the bait and the crayfish, but it was fought a little bit. So to be very clear, we are saying that any live crayfish and live bait is actually restricted. So some clarifications with regards to aquatic invasive species on the regulation front. Education outreach, I'm sure you've seen some of our materials. We have 2 campaigns that we largely focus on: Clean, Drain, Dry Your Gear and the Don't Let It Loose program. And these are really all about moving from just plain awareness to actually making people do something different and changing their behaviors. You might see a little bit of a new look for our Don't Let It Loose. So Don't Let It Loose is actually going national. So all of Canada will be adopting the Don't Let It Loose campaign, and we will be shifting to this new logo. And so a lot of thought went into this new logo because Don't Let It Loose can also apply to terrestrial species. So here we are covering all aquatic and species. The species forefront is what we're trying to prevent the introduction. On the left-hand side is the goldfish. So pet trade industries is being targeted on this left-hand side symbol. But you might notice that it's the act of dumping. So the water bucket implies that dumping part. And you'll notice in the water, there's actually silhouettes of other species that people might not notice that they're actually introducing. So a lot of thought has went into this. It is available in French and English. And the Canadian Council of Invasive Species is probably going to take on the terrestrial side. But we have targets for the pet trade, bait fish, sport fish, plants, and then the live food industries as well. So we've covered kind of all gamuts. As for monitoring, this is where Ron Zerwell helps me support as well as many organizations, including the Alberta Lake Management Society, which Caleb highlighted. We are focused on spiny water flea and zebra quagga mussels through the villagers and adult substrates, as well as monitoring was also added for Eurasian water milfoil in 2019, and thankfully that program is still continuing in 2020 thanks to ALMS support. And so this one specifically is ALMS steering this one. So thanks for the support. So far, we remain Eurasian water milfoil free. We just have confirmed the native ones in many lakes. Watercraft inspections are up and running. I don't have a new map for 2020, but this is what the map looked like last year. The 3 stations at the south border, bordering with Montana, have not opened. We were prepared to open them if our border ever opened, but that date has extended to September 21st and likely probably will keep continuing for most of the boating season. So we are not probably likely opening those stations. Dunmore is still in operation and probably will be until the end of October. Oyen and Rainwhite did not open this year due to staffing shortages. And so Vermilion and Cold Lake will be staggering closures as well. So I think Cold Lake is closing after the September long, so right away here. Dunmore will be up and running until the end of October, and Vermilion will be up and operational to the end of September because of staff leaving. We try and leverage university students, so most of them are returning to school. We have intercepted a record number of mussel-fouled watercraft. So in 2020, we don't have our numbers just yet. We get those at the end of the year season. And so we're on pace to have broken our record. Our record last year, last year and the 2017 was 19. Right now we're sitting at 25 mussel-fouled boats. One still has yet to be confirmed, but we are definitely on pace to have our highest number of mussel-fouled boats. And we attribute this to most folks traveling interprovincially rather than internationally. So lots of folks that probably would have went down to the States were actually coming out west from the east. And this is actually only under 3 operational watercraft inspection stations. So very impressive for that program. Kudos to Cindy and her team for operating under COVID restrictions to keep operational. I also mentioned the canine unit. We have 3 canine mussel sniffing dogs. The left-hand picture Black Lab is Hilo. The German Shepherd is Zeus, and the chocolate lab down below is Diesel. And so they also participate in shoreline capacity inspections, as well as are out there on the watercraft inspection stations also doing that, boat inspections. Response, we still have many calls coming to our 24-hour, 7 days a week hotline. Report a Poacher is basically the call center it's going to. The Report a Poacher gets about 10,000 calls, and our hotline to the aquatic invasive species is probably only at about 500 to 1,000. So very active in getting us in touch with public on a quick response time. Potash registration, we are pursuing to have potash, which is a fertilizer. Saskatchewan is willing to give us as much as we need, and they mine it right in Saskatchewan. They will ship it to us by rail car or truck, whatever we need. And it is 100% effective if we were to get introduction of invasive mussels. So it stops them from breathing. It only targets mussels. So it would affect invasive and native mussels, but the native mussels would definitely recover quite fast. We've submitted this to the federal government for review in February. And yeah, we're just working through some data deficiencies, but hoping that that only takes about 18 months for them to review. So we're hoping that we'll soon have this available to all Canadians. And really it's just the Canadian governments, whether it's province or territory, and so that they have this in their tool belt if they ever have an infestation that they can actually respond to. Flower and Rush, we were very hopeful to get another attempt at addressing the population at Lake Isle. But unfortunately, due to COVID-19 and some delays in meetings, it wasn't happening again this year. But we have some really good relationships built. There's a MLA involved in the project now, so we're hoping to get some more and more attractions, continue those conversations this winter, and get the approvals in place for a large herbicide program at Lake Isle. And just to update those folks that aren't familiar, there's about 17 kilometers of shoreline, various thicknesses across the lake, but it is the largest infestation of flower and rush in Alberta. We're staying on top of our Phragmites. We keep getting more and more locations reported to us. So they are all, relatively small in size compared to what Eastern Canada has. So we're trying to keep it that way. CN and CP seem to be our biggest culprit in bringing Phragmites out west. So we're keeping on both of those railways to stay on top of their locations, but we've had some come to some industrial areas through trucking yards and along some major highways as well. So trying to stay on top of those locations as much as possible as well. We had a huge detection of purple loosestrife late last year out by Wainwright in the Enstrom Pond. The municipality basically uses this pond as a drainage from the town of Wainwright. They had spent millions of dollars in improving the drainage because this was flooding out some farmers. And so they improved the drainage and now connected to this pond to the Battle River. So this purple loosestrife was threatening to be introduced into the Battle River. So we quickly got onto this. The application happened in August, where we sprayed most of it. We'll obviously need to touch it up for a few years. We're probably looking at about a 5-year project where we start just chipping away at the population. So, but yeah, a really good collaborative project where MD of Wainwright did a lot of the heavy lifting with some support, approval support from me, and then the town also jumped in, and the drainage and the landfill was also involved, and some private landowners. So it's a really great partnership out there, and I'm happy to say that it was mostly addressed, but we'll be chipping at the remaining populations. Yellow floating heart was a new find in 2019. It was in just one isolated location in the city of Edmonton, and we worked with the landowner to go in there and hand pull the entire population, and we've been watching for regrowth since. It only has a few minor little resprouts, so it is almost eradicated, but we will keep monitoring that for a few years to ensure that that is indeed the case. And this was purchased ornamentally and introduced on purpose prior to it being prohibited in 2015. So it might also pop up elsewhere where somebody else purchased it and released it unknowingly. Another new find last year was this Chinese mystery snail, a pretty significant shell, up to 6 meters tall. Pretty beefy, nothing looks like it doesn't look anything like our native snails. So we did confirm that it was present in Lake McGregor. We had some picture evidence that it was actually been there for quite some time. I think we have now evidence back to 2015 that it was actually there at the lake. So it's been there for a couple years. So to get a full assessment of where it was, Ron joined me in August with my team, as well as University of Alberta also partnered up. We did some soil sampling around the entire lake of McGregor, trying to find some DNA evidence of where the snails might be. U of A was looking at food webs to see some of the impacts that snails have been having on the food web, and then we can sort of ascertain if it's really critical that we do something about it. But we're really strapped in this species that it has no known control methods, so we're really kind of focusing on containment at this time. So I have drafted a management plan for Chinese mystery snail, since this is the only location we're really wanting to keep it that way, and there is no control options. So that's really the priority, is containment. We want to put a ministerial order on at Lake McGregor that you have to clean, drain, dry upon exit. And with that, we really wanted to provide some tools. So there's a machine called a CD3 machine that offers an air compressor, vacuum, picker tools, brush scrubbing tools. This one is trailered, so it is movable. It is has solar panels, so it's powered externally. And we were actually the first ones in Canada to bring this to Canada. So it is now located at Lake McGregor. We just installed it last week. And so it has a computer on it as well, so I can sign in at any time and see when it's being used and how much it's being used. So really looking forward to getting some stats and seeing how folks are receiving this equipment that enables the clean, drain, dry portion of that ministerial order that's hopefully going to be endorsed soon. We got a weird call from the Canadian Border Service Agency about Oriental weather loach. We did have a scare last year where we thought PetSmart was selling this species, but we were unable to find any in any of their stores. We had worked with their head office to do a sweep of Alberta stores, and they had it on their list that they might have sold it in the past, but no stores reported having any on hand. So CBSA notified me this summer that they might have had a shipment come in to Edmonton. So we quickly followed up with the distributor. A Fish and Wildlife officer went out on my behalf because I don't have any enforcement powers, and actually did confiscate about 40 Oriental weather loaches. So we prohibited these in 2015, and we've done numerous campaigns where we've tried to educate aquarium pet stores. So through his intel, though, he stated that this fish was as common as goldfish. So we're still figuring out how common these fish might be in the pet trade, and trying to get an approach in educating and making sure that there's no location or no occurrences in Alberta. So, but yeah, BC has some infestations where they have been released and established there. So we do know that it is possible. Whether they would overwinter in our winters is probably debatable, but all it takes is just one introduction in a warm location that's kind of sheltered, and we'd find out. So looking to see if we can minimize that through surveying the aquarium industry and seeing how many we have. Onto some species of concern. So these lie outside of our prohibited species, but are definitely worth some effort and control. And then starting with Prussian carp. So this isn't listed as prohibited because the chance of eradication of this species is pretty much non-existent. It is well established in the Red Deer, the Bow River, the Old Man, and the South Saskatchewan. So we are also getting reports. I'm investigating some ponds in the Edmonton area that might also have Prussian carp, but most of them are looking mostly probably like they're just goldfish that have lost their gold coloration and are starting to look like Prussian carp. So still investigating those locations. We do recently got a hotline report that folks were moving live Prussian carp. So working with enforcement folks on addressing that issue of moving live fish as well. So we are encouraging anyone that has Prussian carp or catches Prussian carp to kill it, never return it back to a water body. So this won't control our populations, but it will continue to put a dent in it. We also prioritize this species in the sport fishing regulations guide. So we inserted this infographic where we highlighted Prussian carp, gave a picture of what to look for, some of the impacts. And so we encourage folks to call our hotline if they were catching. So we're getting a pretty good idea of where they are being found and making a map. So we have a better handle on locations where they're being found. We also do get calls of folks wanting to angle them. So there is a bit of a preference to angle them. So working on that as well. Goldfish is still very much an issue. Working with a couple of municipalities to launch some response efforts. We were recently out in a really well-off neighborhood in the Edmonton area and put out some nets, and we were actually pulling in some goldfish. So still work to do. I help support municipalities, but mostly some companies to do some response work and guide the approval work. So this is very much still a problem. Looking to promote this. People seem to still not get that dumping your goldfish is not a great practice. So we need to elevate our education on this front. Whirling disease is still very much active. We are very much concerned about it. They are losing staff capacity in this program. So working with them to try and augment some of the gaps and keep this program going. Whirling disease is very much a concern to our native fisheries, and we are already starting to see impacts in the Crow's Nest Pass area. So looking to change behaviors so that we can minimize the spread from where we're currently finding it, which is very isolated in the water bodies that we have. Crayfish, I mentioned this a little bit earlier on some of the weird and wacky that we're getting. So trying to find out why these things are moving around, why folks like to fish for them. Apparently they are edible. You get a couple bites of tasty meat in there, but also trying to balance the shifts in regulations so that we can kind of balance a really good program from there. So still working on crayfish. We're encouraging reports so that we have a pretty good handle on the crayfish. We are seeing a lot of the native, but there's also other crayfish that are invasive that we'd be very concerned of if we were to find those. And those are like rusty crayfish, red swamp crayfish. There's a few others on our radar for invasive crayfish as well. So encouraging reports of crayfish through our hotline. So if you haven't got anything from my presentation, hopefully you take home the message that I am not in this alone. I am only one person trying to cover 52 plus species all over the province. There are many people behind me that support this. Lots of folks in the ALMS program are definitely supporting it through the Eurasian Water Milfoil surveys, as well as the villager sampling. So tremendous support from external stakeholders, and a quick thank you if you've participated in anything aquatic invasive species. It is much appreciated. So there's more behaviors always to change. We're still getting, bad practices of draining boats right near a drain. We need to be conscious of the water that we're moving and where we're moving it to. Our pull the boat is mandatory. We have a fine around it, but we still approach about 1 in 4 that don't do it. So need to really encourage folks to pull that plug when they're transporting their watercraft. And that's it for me. If we can open it up to questions now.
Awesome. Thanks, Nicole, for your great presentation and update. So we do have a few questions that have come in here. So I'll read the first one out for you. So what are the implications of potash introduction to other native species of invertebrates or fish?
So it would only affect our native mussels. It stops them from breathing as well. So that would be a definite unintended response of the potash. But in other applications, it's been found that they actually respond or rebound quite fast after the potash application. So there's no need to actually reintroduce the native mussels. They can rebound just on their own. That's why we've pursued the potash, is because it's the best candidate with the fewest consequences to other species in a water body.
Okay, great, thank you. And we've got a couple other questions here. So how far north and west has Phragmites made it?
So we've found Phragmites all the way up to Fort McMurray, in the town of Fort McMurray, at a couple locations, as well as in the Grand Prairie area. So it's quite extensive, and we found it as far south as Brooks, Medicine Hat, those areas as well. So there's a few locations, seems to be transportation corridors or industrial yards that have trucking, long haul trucking.
We have another question here. What is the extent of purple loosestrife in Alberta?
So this was our poster child for invasive aquatic species back in the 90s. We had a huge project with Ducks Unlimited where we found that we had a pretty big problem of purple loosestrife in Alberta. We partnered with Ducks Unlimited for about 10 years, and we did some major eradication projects. Unfortunately, funding ran out. They weren't quite eradicated in all locations, but we are seeing just a few locations now. Wainwright being one of the bigger ones that we've seen recently, but usually they're pretty small. Purple loosestrife is easy to address if you can catch it when it's just a small little patch, because it can be dug out, as opposed to something like flower and rush or pale yellow iris. Those ones are tricky to dig out.
Thanks for that. Another one coming at you. So were any of the 25 mussel-fowl boats that were intercepted non-motorized?
Good question. We have in the past intercepted some kayaks and some dragon boats that were fouled, but I believe this year, I think they were all motorized. But we have proven to other jurisdictions that those non-motorized ones are a threat, even though other jurisdictions had sort of, ignored them in their watercraft inspections. So we have taken the approach that any watercraft, whether it's motorized or non-motorized, needs to be inspected because of those past cases.
Great, thanks, Nicole. We have a question here about the dog. So have the dogs sniffed out invasives, and if so, how many incidents?
So they did detect a boat, so it had evaded the human inspectors this year. So it was solely up to the dogs that they actually intercepted one of those boats. So they're exceptional at sniffing things out. We've tried adding some other species to their repertoire. So we're helping other programs like the wild boar program based on their ability to sniff out wild boar scat in winter to track down populations. They're also trained on a unknown, very small, minuscule plant, hard to recognize, a plant called Thesium in the Calgary area. So they are trained on multiple species, as well as they do the shoreline, so they're able to just find the larval stage of mussels in water.
Great, thanks, Nicole. And we've got another question here. With the Crow's Nest Pass being a popular recreation and angling, specifically trout destination, is there currently a focused effort on this region for whirling disease or plans to educate anglers and rec users?
Yeah, so we have signs in place trying to notify anglers that may not be familiar with whirling disease that they are entering that whirling disease infected area, and that they should be clean, draining and drying all gear that touches. And we have interactive signs basically outlining what that means to clean, drain, dry. So we're hoping that will help reach those folks. We have, a whole program that was supporting that education piece as well up until recently.
Thanks, Nicole. I think there's no open questions. If you have questions, just a reminder, use the Q&A function. We probably won't spot you if you just put up your hand. I do have a question for you, Nicole, about the zebra quagga mussel situation in other provinces right now, and if you know how other provinces are faring, Saskatchewan and Manitoba maybe.
Okay, yeah, so Manitoba has had some new detections closer out west to us along their western border. So they are still very much trying to contain the locations that they have. They have an exceptional program. They have a huge enforcement that encourages decontamination of all boats when they're exiting a water body. So they've done amazingly with what they have. trying to stop those boats from coming out west. We are still intercepting some, obviously, because we have 25 so far this year, and most of them are coming from either Ontario primarily and a few Manitoba. So much like our program, we are only as good as our best defense, and unfortunately our inspection stations can't be open 24 hours, 7 days a week, much like their program. So we combine efforts quite extensively. So if they're not caught in Manitoba, we're hoping that they're getting caught in Saskatchewan. If they're not getting caught there, hopefully Alberta's catching them. And if we're not, then hopefully BC's catching them. So we have a really good network amongst the provinces that don't have it, and we communicate quite often.
And maybe what's the biggest limitation to the inspection program each year? Is it a funding situation? And is there some way that folks watching this presentation can help or advocate for, you know, a more robust program?
Yeah, funding is probably the main factor. Yeah, it's basically funding. It's a huge undertaking. If we were to set up one inspection station in Ontario, we could probably catch all Western traffic. Once we get past Manitoba, then the highways branch out more so, and then we have multiple points of entry. So we can't be at every crossroad across the province. So we pick and choose the main highways as best we can, and we have a pretty good stat space for which ones are most effective in catching mussel-fowl boats. So we prioritize with what resources we're given to make sure that we have the best coverage for Alberta as we can.
Just got in another question for you. So in general, are invasives coming from the east or the south?
Primarily this year, because of international borders have been closed, it's been mostly out east, and that sort of has been the trend even with international borders closed. Most of our boat traffic is from out eastern Canada that we found. Other boats from south states like Montana, Idaho, Wyoming don't have mussels either, so we're pretty low risk for any boats coming for a boat trip up from the US.
And do you have any thoughts on the future of the AIS program in terms of maybe using more eDNA techniques, or is kind of the microscopy the best method that we have right now?
Yeah, so other jurisdictions haven't quite made the shift to eDNA. We're still looking for the science to really make that a robust, reliable science. It's a good indicator tool right now, but it's not as reliable as the microscopy of villagers. So there might be advancements in short order, but right now we're relying on the microscopic analysis of water samples.
Okay, are there any other questions from folks on the Zoom call?
I have a question. For the potash application to control mussels, would that be a spot application to where adults are identified in a lake, or would it be a distributed application across the lake to try to capture the villagers as well?
So it depends on what the detection or the report indicates. So in the example of when they detected mussels in Lake Winnipeg, they thought it was just in the South Basin. So they isolated the South Basin with a curtain and were able to put in potash under a research authorization, basically, from the federal government. It was 100% effective. They killed all the mussels in the South Basin. But through further monitoring, they found that it actually had broken through where they thought they had a line of defense, and it was already established in the North Bay. And as the ninth, I think it's the 9th largest lake in the world, Lake Winnipeg was too huge to do an entire treatment. So it would depend on what the detection was in Alberta. Hopefully we could isolate it to a small area and just treat that small area. But It may be as big as an entire lake, depending on how fast we get it detected.
Okay, I don't see any other questions right now, so we'll just say thank you to Nicole for putting this presentation together for us. Really appreciate it, and of course, if there's any way we can support you in monitoring activities, always let us know. We're really happy to help.
You're a fabulous stakeholder that helps me out tremendously, so thanks.
And of course, also thank you to Caleb, our first speaker this morning. I think that was actually Caleb's first ALMS presentation, so a really great job, and he knows the programs really, really well. So I'll just mention that the webinar series is happening every Friday in September. Next Friday, September 11th, we have two presentations, one on microplastics in freshwater. That's from Dr. Michael Rennie from Lakehead University. He's also the Canada Research Chair in Freshwater Ecology. And following that presentation, we have Dr. Laura Johnson from Heidelberg University, and that's in Ohio, and she's going to be talking about agriculture and the reeutrophication of Lake Erie. So the Zoom link that we're all on today is going to be the same link that you'll use to sign into next Friday's webinar. And I believe you're going to be sent a kind of standardized questionnaire after today's webinar. I think Janine mentioned that, so maybe keep an eye out if you do get a questionnaire. Yeah, and also in the email that you got after registering, there's a note at the bottom that if you want to support ALMS and future events like this, you can always make a charitable donation to ALMS through Canada Helps, our link on the Canada Helps website. So with that, I just want to thank again all of our speakers and all of our attendees for joining us today. And of course, thank you, Janine and Jenna, for helping me put all of this together. Very much appreciated.
No problem. Have a great day, everybody.
Okay, bye everybody.
Thank you. Bye.